Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Companion Diagnostics and Clinical Development in Oncology, a Statistical Perspective. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by using that chat panel and send me a message, and I'll make sure I get back to you with any answers. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank IDDI, International Drug Development Institute, who developed the content for this presentation. IDDI is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Massachusetts, Raleigh, North Carolina, and San Francisco, California. Now it's my pleasure to introduce you to your speakers for today's event. And first, I'd like to introduce you to Elizabeth Court, Else, Principal Statistician in the Consulting Services at IDDI. Else is IDDI's expert in analytical and clinical validation of IVDs. She has a strong background in assay development combined with 10 years of experience as a statistician for biotech and diagnostic industries. She has a long-standing interest in Alzheimer's disease biomarkers. Her areas of expertise include expert consultancy in biomarker and IVD development and validation, and expert consultancy in clinical trial design. Now it's my pleasure to introduce you to your second speaker, and that is Dr. Everardo Saad, Medical Director at IDDI. Everardo has over 15 years of experience in medical oncology and clinical trial designs. He did his fellowship in the medical oncology at the University of Texas MD Anderson Cancer Center in Houston. After practicing for several years, he shifted his professional career towards education and research in medical oncology, and has a special interest in clinical trial methodology, the assessment of endpoints, and the development of novel therapies for cancer patients. And now it is my pleasure to pass over the controls and the mic to our first speaker, and that is Everardo. So Everardo, you may begin when you are ready.
Thanks a lot, Sonia. Hello to everyone. Thank you for attending this webinar. So our plan today is as follows. I'm going to do a relatively short introduction where I'll talk a little bit about precision oncology, what we mean by this, and the relevance for the topic at hand today. And then just some definitions and current examples of biomarkers. And then Else will come and tell us the main steps towards validation of IVDs, and then tell us statistical aspects and pitfalls that need to be taken into account when validating companion diagnostics. So let me start by giving a definition of precision oncology. I don't think there is let's say, universally accepted definition, but usually people refer to this as the attempt to give the right treatment to the right patient at the right time. And what is implied here is that we need to use targeted therapy and biomarkers, and this is particularly relevant in oncology. The way I see it, there are at least three meanings that can be denoted by definition that I just mentioned. There are subtle differences between them, but I'll give examples to show what I mean by these three definitions. There is some overlap between them. So one is to use molecular features to select evidence-based medicine, and if it's oncology, that usually this means molecular features of tumor tissue. Then the second one is sequencing tumors to select among existing therapies. And then the third one is choosing treatment based on predictive molecular features. Like I said, there is an overlap, but I think there is a subtle difference between these three situations. So the first one is to use molecular features to select evidence-based treatment. And this is what we would do, for example, just two examples here. One is a drug that targets a very specific molecular abnormality, namely in tract fusion. And this drug is active in several tumor types. And so this is a situation where you have to check for the presence of the biomarker and use a treatment that is approved for this biomarker. And likewise for this situation in lung cancer, non-small cell lung cancer, where if you have specific alterations such as ALK molecular alterations, this drug is superior, for example, to chemotherapy, at least in terms of progression-free survival or PFS. So regardless of whether we're talking about evidence that comes from, for example, a single-arm phase 2 trial or a large phase 3 trial, in both situations, we're looking at the available evidence to choose a certain therapy based on molecular features of the tumor. Now, there are situations, like I said, similar, but in this case, we have patients, for example, that may have various different molecular alterations, and we may look for these alterations, for example, using next generation sequencing or other types of these multiplexed types of assessment, and then look for whether there may be some existing therapy, whether approved or not, that can be used for this patient. So in practice, oncologists do this more and more in their daily practice. So there is a situation, for example, of an observational study in which patients with lung cancer who got targeted therapy matched to their alterations had better outcomes than patients with similar alterations not getting matched therapy, or patients without such alterations. But this is observational study. We know there may be a risk of bias. In fact, to the best of my knowledge, this is the only randomized trial of this approach, and there was no improvement by using this kind of targeted therapy. I'm not saying this should not be done, but it's still open to question whether looking for whatever molecular alteration is out there and trying to match therapy to patients, it's not proven, at least with high level of evidence, but it's a way to practice precision medicine. And then the third way is to choose treatment based on what we might call predictive molecular features. And there are two typical examples here. On top, it's non-small cell lung cancer, EGFR activating mutations, and in the bottom, it's colorectal cancer, KRAS mutations. So we know that activating EGFR mutations are predictive of benefit from TKI inhibitors such as jeopardize. So if you have a patient with the presence of the mutation, jeopardize is better than chemotherapy. Conversely, if you have someone who does not have the mutation, chemotherapy is better. So this is what we might call a qualitative interaction, or this is predictivity at its best. So you can really select for different patient populations regarding the presence of the mutation, you can select between targeted therapy or chemotherapy. And this is similar, for example, for antibodies against EGFR in colorectal cancer. If you have someone with a mutation, these drugs do not work very well. If KRAS is wild type, there is evidence of benefit. So this would be more of a quantitative interaction. So we come to the point that we have to define what a biomarker is. And nowadays there is a very useful document that I suggest for those of you with an interest in this topic, written by FDA and other people. So they define a biomarker as any characteristic that can be measured as an indicator of normal biological processes or pathogenic processes, or even response to an intervention, for example, certain treatments. And these features, these characteristics may be molecular, histologic, radiographic, it doesn't matter. So in fact, anything can be considered a biomarker. It's a very broad definition. Obviously here, we're going to be focusing on molecular biomarkers. And so there are several different categories of biomarkers. And the ones that are more relevant for us today are the ones here in bold type: diagnostic, prognostic, and predictive biomarkers. So let's just take a quick look at what these mean. A diagnostic biomarker is any biomarker used to detect the presence of a disease or condition of interest, or sometimes to identify subtypes of the disease, some groups of patients with a specific feature. So this is not so relevant for precision oncology, but some of the concepts used in diagnostic biomarkers are indeed relevant, and that's why we left the definition here. Then we have prognostic biomarkers, which are those used to identify the likelihood of a certain clinical event, which may be response to therapy, the duration of survival, or any other event of interest. So a typical example in oncology, the same EGFR mutations that I talked about are considered prognostic in non-small cell lung cancer, because patients who have activating EGFR mutations tend to have a better prognosis than patients without the mutation, even before the availability of specific targeted therapy. And finally, what's more relevant to today's discussion is the notion of predictive biomarkers, which are those used to really select patients based on the presence of biomarker. And like I said, when there is what we call interaction, in the sense that one treatment subgroup does better with one treatment, and conversely, the other subgroup does better with another treatment, or has no benefit from the first treatment, that's when we talk about predictivity, and that's the ideal situation for precision oncology. Now, validation of biomarkers is something that Els will discuss in more detail, but there are three concepts that are often used, which are analytical validation, clinical validation, and the notion of clinical utility. So analytical validation is a series of analytical steps to determine the performance of the test or of the assay in this case, such as sensitivity, specificity, predictive values, etc. So this has to do with the assay itself. Then, if we can establish that the test identify or measure or predicts some concept of interest, for example, prognosis, this is what we call clinical validation. And then the clinical utility of a certain test or biomarker requires going one step further and showing that the use of that biomarker, for example, leads to improved health outcomes. And so the validation entails all these steps, and it's good to be aware that at some points people are talking about validation in a more broad way, and it's good to have in mind what step of the validation is being taken into account. Now, oncology, unlike what we might think, is not as rich in validated biomarkers as we might think, at least if we think about a biomarker in a generic way. I'm talking here about the molecular alteration itself. I'm not talking about the specific IVD or product out there. Then there are many more, as I will show in the next slide. But if you only think about the alterations for which there is targeted therapy, whose selection is based on the presence of the alteration, there aren't that many. And this is an almost complete list of all the available biomarkers in oncology. I don't need to go into every single one of them, but these are the ones that are out there for treatment selection based on let's say, label indications. So this is another type of information I'm showing here. I just copy and paste the conclusion of a study in which the authors tried to find whether the available biomarkers in oncology, the predictive biomarkers, were really tested based on, let's say, the best possible statistical evidence, which is when a treatment by biomarker interaction is really demonstrated, such as the examples that I gave of EGFR mutations and KRAS mutations. And they came to the conclusion that in many cases this is absent. So there are lots of biomarkers for which we don't have clear proof of interaction, and in many cases, perhaps rightly so, the patients with biomarker negative tumors have not been investigated. I think when there is a very strong rationale for using a certain drug targeting a specific biomarker, there is more reluctance, more difficulty in really proving that there is an interaction, simply because studies are exclusive or enriched for patients with positive biomarkers. Now, a final definition that I need to give is that of companion diagnostics, which is defined by the FDA as a device, an in-vitro device, an IVD, or an imaging tool, something we won't cover today, that provides information that is essential for the use of the corresponding therapeutic product. And that's why they're called companion diagnostics, because they walk hand in hand with the therapeutic product of interest. And so the FDA has this list, which they update periodically, available on this website. And the list, I took a look at it recently, and it contains 44 approved companion diagnostics, 43 of which are in oncology. So this gives an idea of how oncology is spearheading, let's say, initiatives in precision medicine. The only one that is not strictly in oncology is in oncology for thalassemia, and that's, by the way, an imaging tool. And of the 43 oncology, or of the 44 companion diagnostics, 10 are dedicated to one single biomarker, namely HER2. And there are additional IVDs which are approved, which are trying to find several different types of molecular alterations, including HER2. So if you only think about HER2 as your marker of interest, there are not only these 10 products, but additional ones which are multi-gene or multi-molecular alteration, let's say, types of IVDs. So this is what I plan to show as an introduction, and now I'm gonna pass the microphone to Els.
Yes, thank you, Everardo, for the nice introduction. So I can skip the definitions. I think the scene is already well set. The one thing I want to say before diving into the validation process is that when I will be talking about validation, it's really in the regulatory sense. It's validation of a test of an IVD as part of the regulatory trajectory towards the registration and marketing of the product. So validation, it marks the end of a development phase. After several cycles of development within an assay development setting, an assay will be locked down, means all the design features will be locked, will be well described, and then the validation will start in 2 steps, the analytical validation followed by the clinical validation. Now, maybe one thing that is important to know before the validation, so part of the aspects that need to be locked down is a very important one when we discuss companion diagnostics, and that's a cutoff. Because most biomarkers and assays, they do measure a quantitative, on a quantitative scale, it's a quantitative measurement, but in practice, the biomarker will be used in a dichotomized way. So with a cutoff, we just make it positive or negative, and this cutoff is part of the IVD and should be locked down and should be defined prior to validation. Now, we are used to see in papers and so forth means and standard deviations, and we always know that these are estimated with a certain associated uncertainty, but the same is true for cutoffs, but this is not always described. So when we define a cutoff, we always report it together with its associated uncertainty, just to make sure that the audience understands how precise the cutoff was estimated. And this uncertainty can be quantified with a confidence interval, and this confidence interval is a kind of gray zone in which you can see the biomarker values as inconclusive. Because if you would set up the same type of cutoff study, same sample size in the same population, you could end up with a cutoff within this confidence interval. So that's why the values within this interval are often seen as inconclusive. Now, I think most people agree that a cutoff needs to be estimated in a precise way, but the question to what is sufficiently precise, it's not always easily answered. At the bottom, I have a formula that can be used for the size of a sample size study, but this is really in a diagnostic setting when you can relate the precision of the cutoff to the desired precision of your later clinical sensitivity and specificity estimates. But in the companion diagnostic setting, this might be less relevant, and so it's often more intuitive approach that's taken to what is sufficiently precise. And that could be that the width of this confidence interval is not exceeding a certain proportion of the full clinical range, so the full range of measurements possible. And the one that I might like better is the second one, because it's really related to the intended use. It's to set a maximal proportion of subjects with an inconclusive biomarker value, so with a value within this confidence interval. Meaning 50% of your measurements fall within this confidence interval, then of course most people will agree that this is not sufficiently precise, but then what is precise is debatable, of course. Maybe something related is not only the cutoff should be fixed or locked down prior to the validation, the same holds true for a signature. So if we are not talking about a single biomarker, but about a signature that's built up of several biomarkers, we need to define which set of biomarkers will be part of the signature, which algorithm will be used to combine the information, and which cutoffs will be set to this biomarker score, or if it's a tree-like algorithm, which cutoffs will be used in the algorithm. I want to point out that for the moment, among these companion diagnostics, there's no true biomarker signature yet. I would say there are, of course, companion diagnostics that are multi-gene, that measure, that are based on next generation sequencing and measure several genes or several alterations within the same gene, but that's not the same, of course, as a signature. Now, when we say this has to be locked down prior to validation, this is not because there's no statistical methodology available to set the cutoff or finalize this signature during the validation process, but it's more a restriction that's posed upon us based on the regulatory approval. For now, the regulators, they tend to, they want to see the definition of the intended use population before the trial is started. And of course, it's the cutoff that sets the intended use population and will define which patients are part of this population and which are not. The other reason why it would be difficult for a companion diagnostic to derive the cutoff during the validation is that the analytical validation would need to be done together with the clinical workup for the drug, and the analytical validation, as I will show, relies on this cutoff. Here, the ideal situation for the validation process of a companion diagnostic, and here on top you see the drug that is taken through the different development stages, from the preclinical to the early development stages, phase 1, 2, and 3, coming to the regulatory process and can be available on the market. At the same time, the assay is developed that will become the companion diagnostic. It will undergo analytical validation, and then later on clinical validation and regulatory submission and market approval. Now, for each phase of the clinical development, you can use, of course, a different assay. As long as the assay is locked down, you can use a different version, and the chances are very few that you would have already your final version of the companion diagnostic ready when the drug is only in phase 1. The question often arises, okay, if we can use these different assay versions, do we need to go through the full validation process for every new version that's used in the clinical trials? And the answer is there need to be some new validation for each version, but the level of validation is not the same. So in the early phases, it's more kind of an exploratory type of validation, whereas for the pivotal study, where the pivotal study for the drug, there you would have really an advanced analytical validation, and this would consist of many different validation studies that rely on the CLSI guidelines that FDA recommends you to follow for the analytical validation. Or, I mean, in some cases, for some type of assays, this is not possible, but then it will certainly speed up the registration process if you can provide a rationale why you are deviating from those. I want to point out that and I think this is very well known by the assay developers, but maybe not by the people involved in the drug development process, that this analytical validation result, it's a substantial part of your FDA submission package. So it's both the analytical and the clinical validation results that will be really scrutinized together by FDA, and it's by no means something that is done in the lab and stays in the lab. I can't cover all the different analytical performance characteristics. I will just today cover two of the most important ones, precision and accuracy. These are performance characteristics that are important for most assays and certainly for a companion diagnostic. So precision meaning that your measurements are closely together, and accuracy meaning that you are on target, like here and here. So as said, precision, it's about the closeness of agreement between replicate measurements on the same object under specified testing conditions. It's really about testing the robustness of the assay. And the testing conditions will define the different types of precision. It goes from repeatability, in which you will keep the testing conditions as similar as possible, to reproducibility, in which you try to include as many sources of variation as possible. That will include, for instance, different labs, different instruments, different operators, and so forth. Now, what is particular for a companion diagnostic, because it's most likely a qualitative test, just discriminating between positive and negative cases, is that we will express precision in this case as a percent agreement with the majority rule. As I said, just to evaluate how close the different measurements of the test are together. And in this case, reporting the precision as a standard deviation or a coefficient of variation, what is often done in assay development, is more for information only, because this standard deviation will not tell you a lot in terms of how precise the test is in terms of the intended use. And that's the reason why this qualitative precision, as I would call it, has to follow different guidelines and different rules for analytical validation. In the CLSI guideline, there's the concept of an imprecision profile, and you will be asked to, for your test, to define what is the C5 and the C95 interval. This interval is limited by the C5, C95 concentration, and these are the biomarker levels that result in 5% or 95% of positive test results. And a good test, a test with a good precision, will have a narrow range, because in this range, the test will be considered as too imprecise to be reliable. So, and this is how such an imprecision profile looks like. You have here on the y-axis the percent positive result. So for the red, for the test in red, you see here the C5 concentration here, and you see the C95 here. And so this interval is smaller than for the, is narrower than for the blue curve, meaning that the red test has a better precision profile than the blue one. Now, I just said that for a qualitative test, it's not informative to provide a standard deviation or a percent CV. But of course, the qualitative and the quantitative precisions, these are two sides of the same coin, and one can be derived from the other. So if you give FDA or another regulatory agency, you present them with one of the two, they will be able, of course, to translate it. But of course, it's much more important to show them for a qualitative assay where exactly your C5 and C95 range is. The second analytical performance characteristic is accuracy. And this is also often expressed as a kind of agreement. In the analytical validation of a qualitative test, there's a lot of agreements, but it's not always between the two same targets. So the accuracy can be expressed as positive and negative predictive value. The positive predictive value being the probability that a patient with a positive test is truly positive, and negative for the negative predictive value. So here we're really trying to assess what the agreement is between the candidate, companion diagnostic, and the reference standard. Here it's important to say that the common design for analytical accuracy is a design with single measurements for the candidate and the reference. And in fact, if they don't agree, it's very difficult to disentangle between a systematic and a random error, or between accuracy and precision. So there will always be some imprecision involved in this type of study. So this was just a snapshot of some analytical validation characteristics, and let me now go to the clinical part of the validation. So in the ideal case, the pivotal study that will assess the clinical value of the drug will also test the clinical value of the companion diagnostic simultaneously, and so we come to the biomarker-driven clinical trial designs. A classical trial design will have more broad eligibility criteria, will see the treatment effect as the average outcome, whereas for the biomarker-driven trial, we will be really looking at the treatment effects within the biomarker-defined subgroups. There are so many different biomarker-based trials. I'm just showing you the two ends of the spectrum. So the one end of the spectrum is an all-comers interaction design, in which during the screening, the patient, the biomarker status is defined, and within the biomarker negative and biomarker positive, the patients are randomized within each stratum and are getting the standard or the experimental treatment. Treatment effects are estimated in both strata, and the study is powered to really pick up this interaction, so really to pick up the difference in treatment effect between the strata. This would be a design that is, statistically speaking, the most robust. It would show the predictive value of the biomarker, but as it requires an unacceptably large sample size, this is not often implemented. At the other end of the spectrum, we have the targeted or the enrichment design, in which during screening the biomarker is tested and the biomarker negative patients are excluded and only the biomarker positives are randomized over the treatments. Of course, this is a much more efficient design. It avoids potential harm in the biomarker negative subset, but it does deny this subset a potential benefit of the experimental treatment. And so this design is really mostly used when there is a very clear relationship between the biomarker and the mechanism of action of the drug. Taking one step back, so not only in the pivotal studies, we will see many biomarker-driven designs, also in the earlier phases. And again, here, with the strong relationship of the biomarker and the mechanism of action, we see that often the biomarker-positive patients are included only, and we see many single-arm designs. A very typical enrichment design are the basket trials, type of trials we see a lot these days in precision oncology. And it's a type of design in which different types of cancer are grouped or are put in together as long as they have this one common targetable mutation. And in 2017, it was, I think, was the first, what we would call the first basket approval by FDA, or maybe better, an histology agnostic approval, where pembrolizumab was approved in many different solid tumors. So you can see here all the list of the solid tumors, as long as this tumor had this biomarker, so a microsatellite instability high or a mismatch repair deficiency. So the microsatellite instability, it's tested with a PCR test, and the mismatch repair deficiency, it's tested with an immunohistochemistry test. This first approval, this first basket approval has led to some problematic clinical utility, and to maybe frame it positively, it has shown that a companion diagnostic is really, might not be such a crazy concept and might be something we need in precision oncology. Because here the patients were tested with local lab developed tests, with the two different tests, and it was very difficult for the treating physicians in clinical routine afterwards to decide whether his or her patient was eligible for this treatment or not. If the patient was negative on microsatellite instability, should then the patient be retested for the other tests, just to make sure that we don't miss out on this treatment, or would this be not needed? So there was a lot of discussion about this. But now in a more recent basket approval, this was in June this year, it was for the same drug, pembrolizumab, again in solid tumors, but now it was for another biomarker. It was for tumor mutation burden. And now together with the approval of the drug, so extending the label of pembrolizumab, there was also the approval of the companion diagnostic that was developed at that time. So now it's clear that the patients need to be positive with this test to be eligible for this part of the labeling of pembrolizumab. The basket trials that I just discussed, they grouped all the different indications into one cohort and then investigated whether there was activity of the drug. But what we see is that many of the sponsors we work with, they would like to see within this basket trial different cohorts defined by these different histologies. And they want to test the effectiveness of their drug in each of these indications. So it means that the basket trial is made up of many different single-arm trials. Now, I recently listened into the webinar of a statistician, and he said, well, we don't need any statistical rationale for the design of this single-arm trials. Well, we don't agree to that. I mean, we do know that this type of phase 2 trials, they don't give a final answer to whether the drug will work or not, but we would like to see a proof of concept, of course. There are many different designs that could be used for these different cohorts of single-arm designs, but I'm not going into this. Everardo and I, we did a webinar last year on expansion cohorts, and there we discussed about single-arm designs. Also, very importantly, why we think that even in this case, it would be wise to randomize. But for the audience, the people in the audience that are interested in these different designs, we would refer them to that webinar that is still available. One thing I would like to say about these enrichment designs is that it's common knowledge that the treatment effect that we want to pick up, that will drive the sample size. If we want to pick up an effect in let's say an increase in response rate of 20%, that will not need the same sample size as an effect of only 10% absolute increase. Now, what I would like to add here is that this treatment effect is in turn influenced by the analytical performance of the test you use to define the intended use population. And that is, of course, because in an enrichment design, you expect that in the biomarker negative group, there will be no treatment effect or a lower effect. And if your test is not 100% accurate, no test will be 100% accurate, then you are diluting the treatment effect with patients who don't benefit, right? You cannot, this dilution effect is based on two quantities, one being the ratio of the treatment effects in the true biomarkers, positive and negative. This is something you can't influence, of course, but what you can influence is the analytical accuracy of your test. So this just to say that spending an additional few months in the lab to improve on the analytical performance of the future companion diagnostic, it will pay off in the later phases during clinical development, and of course also during for the clinical utility in the end. Having the simultaneous development of the drug and the companion diagnostic, that is the ideal situation, but in practice this does not happen a lot. Often the final version of the companion diagnostic is not ready when the pivotal trial is run for the drug, and then you will have to do a bridging study afterwards. So it will be an additional study that is needed before you can market your diagnostic product. The clinical trial assay is then the name we give to the test that was used, and you will have to bridge the results to your companion diagnostic. Now, of course, the agreement between those two tests is important, but it's more than that. FDA sees this bridging problem as estimating the drug efficacy in the companion diagnostic subpopulation. So you really have to show that the clinical benefit of the drug would have been maintained in case you would have had already the companion diagnostic at the time of the pivotal trial. There are many different statistical approaches to go about for these bridging studies, and choosing the right approach really depends on the availability of the clinical trial samples and also on the pivotal trial design. Was it an all-comers design or an enrichment design? And I will remain in the example of the enrichment design to show you what this bridging study could look like. So in the end, what it is that you will now be shifting the intended use population of the drug. When the drug was approved with the clinical trial assay, and in an enrichment design you only included the biomarker positive patients, then this is your intended use population for which you have a treatment effect. Now you bring in a new test, a companion diagnostic, and you will say that you will use it to identify the intended use population. So this is this population, right? And it means that those patients are no longer part of your intended population, but these patients, they do belong to this new population, but you don't have any information about their treatment effect, right? So that's in essence the situation you are facing and you have to bridge. Now, although it's not about agreement, it is helpful to define a minimum level of agreement between your companion diagnostic and your clinical trial assay, and that you can do by creating a weighted average of the treatment effect for the patients that are positive with both tests, and here an assumption for the treatment effect for the patients that are, for which you don't have a clinical outcome because they were no part of the previous population, and then you weight it by the agreement. So here the probability that when your companion diagnostic is positive, what's then the probability that also your clinical trial assay was positive? And here 1 minus this probability. And if you plug the values in, so you have an estimate here and you plug in different agreements, you will see that from a certain disagreement onward, your treatment effect will no longer be significant. And that would then be the minimal level of agreement you need to proceed with this bridging. Now, if you don't have samples available of this pivotal clinical trial or not sufficient samples, or you don't have access to them, then you have to rely on an external concordance study. This will be the case for a follow-on companion diagnostic like we might see more often now. It's a kind of me too product when there is already a companion diagnostic in the label of a certain drug, but you also want to market a diagnostic for the same indication. And in this case, we can only do an indirect assessment of the therapeutic efficacy through agreement between this follow-on and the original companion diagnostic. Here, of course, the assumption of transportability or the external validity becomes important, and it's a very tough exercise, I think, to do. So this is what I had prepared for today. But what I would like to emphasize is that precision oncology relies on companion diagnostics with excellent performance. We shouldn't forget that this performance also includes analytical performance, as this drives the later clinical performance. And although that I'm well aware that the development and the validation of a biomarker is about much more than statistics, we should also not forget that statistical rigor is needed throughout this process. So thank you for your attention, and then I'll give the screen and the word back to you, Sonia.
Okay, thank you very much, Els, for that presentation, and Everardo. Thank you very much. I hope everyone found it very insightful. Now I would like to invite our audience to continue sending in their questions or comments right now using the questions window for the Q&A portion of the webinar. I've already received some questions, so I will start with those. So Els and Everardo, are you ready?
Yes.
Yes.
Oh, okay, here we go. Here's our first questions from our members of the audience. What is the added value of a BM in terms of success rate of the treatment? Has this been reviewed retrospectively?
Okay, yes, so assuming BM is the biomarker, yes, well, it's a very good question. I don't know if it has been reviewed retrospectively, and I'd really like to know. To the best of my knowledge, I mean, the question about the added value is, I think it depends, how you answer it depends on, whether you're really talking about precision oncology. Because if you are, I don't think you can talk about it without the biomarker. But I know that in real life out there are many targeted therapies for which there is no biomarker. And so in that in that sense, these drugs have been very successful. I can give some examples. For example, bevacizumab is one example. Proteasome inhibitors in myeloma, for example, HDAC inhibitors. So these are drugs that are highly targeted, but we don't know what biomarkers are relevant, and much less which ones are predictive. So a drug may indeed, and maybe a more recent example with immunotherapy, it's still unclear which one is the correct biomarker for many of the PD-1, PD-L1 inhibitors as far as predictivity. So a drug can be successful without a biomarker, but if you're talking about precision oncology, I guess a biomarker is needed by definition. That's my take, and I don't know about if someone has looked at this quantitatively to see, let's say, correlate the chances of success with the existence of biomarkers, but it's a very good question. I unfortunately don't know the answer. To the best of my knowledge, no. Else, do you want to complement that? No. Okay.
Okay, thank you. That was a very thorough answer, I think. Okay, here's our next question. What's your perspective in terms of how to select the appropriate biomarkers to go through analytical and subsequently clinical validation?
So it's about how to select the best biomarker to take forward for the validation process. Well, not an easy question. It's really about biomarker development, right? I don't know. I don't know what to say. I think you'll have to test many different ones, but I don't know if there are.
Yeah, no, I cannot add much. But I guess the question is perhaps if you have many potential biomarkers, which ones do you take forward? Do you take all of them, or how do you select? How do you?
I would at least take all of them in the first phases of the clinical development. Then, of course, if you're still doubting about what would be the best biomarker, then the enrichment designs might not be the best because you don't have a lot of evidence yet on where it would work. So that you gather enough information on a broader population so that you can analyze the data of your earlier phase trials to make this decision for the pivotal design, I would think.
Okay, well, thank you very much for that answers. Here's our next question. Apart from the statistical design, are there other statistical aspects to be considered in the protocol for an analytical validation study?
Yes, well, I think apart from the design, of course, we have to make sure that there's a well-defined claim in there. We cannot go in saying we will test precision and we'll see where we end up. A claim should read that we want with 95% confidence, we want a precision of this or this level. So the claim should be well defined in a statistical way.
Okay, thank you very much for that. Okay, we're going to go to our next audience question, and that is, when should a sponsor developing companion diagnostics start interactions with the regulatory agencies?
Yeah, then I can only say the earlier, the better. The agencies, they are used to these interactions and they prefer you to come early and to ask all your questions. Even for the analytical validation part, like for the precision, I have been involved in sessions where there's like 2-3 back and forth communications to come to the final design. So I can only say that the earlier, the better.
Okay, thank you very much for that. Here's our next question. Can different versions of an assay be used in a clinical trial?
Well, it is allowed, you can do that, but it will make your life very difficult. And if it is a pivotal trial, it's not something that I would recommend.
Our next question. At what point in the life cycle of an assay development does a sponsor decide to explore a complementary pathway instead of a companion DX pathway?
Yeah, that's the complementary diagnostic. Well, I have to say honestly that I've never understood the concept. It's a concept saying that it's a companion diagnostic, but not quite. So I think if you if you really want to have a test that will define the intended use population, then it is a companion diagnostic per se.
Everardo, do you want to add to that?
No, I don't know. There are some subtle differences between complementary and companion, and
yeah. Well, for me, they are so subtle that I don't. I've never been involved in or never had any sponsor that asked me about.
But it's probably fair to say that from a statistical perspective, there wouldn't be.
There wouldn't be a huge difference. I think it's just that the bar would be put a bit lower when we talk about complementary, because, yeah.
Oh, I see. Okay, perfect. Thank you very much for that. Well, this looks like it might be our last questions. We do have a few more minutes if the audience wants to send in some more questions. But here is this one here. In a biomarker for prediction of therapy efficacy, how do you determine NPV and PPV?
Well, but I talked about the negative and predictive value in the analytical sense, right? So we cannot do the clinical NPV and PPV per se for a predictive therapy. So I'm not sure. Maybe this was what the question was about.
Yeah, the question is not very clear to me either. So how to determine the negative predictive value and positive predictive value in a biomarker for prediction?
Yeah, so this is part of the clinical validation. So these are clinical outcomes for a diagnostic biomarker, for a diagnostic IVD, but I wouldn't say not for a predictive biomarker. We do use these concepts in the analytical validation, where it's about the agreement between the companion diagnostic and the reference.
Okay, thank you very much. It looks like we have one more question here from the audience members. Let's see if we can squeeze this one in. In your experience, has the FDA been accepting of using a subset of CT samples in a bridging study if all CT samples are not available?
Yes. Yes, I think it would be the exception if all the samples would be available. It is, of course, debatable on what is a sufficient proportion of samples to proceed with the bridging study. I have been involved in studies in which 80% was seen as sufficient. And I don't know if you can go below, but 80% does not seem to be a problem. Of course, the larger the proportion of unavailable samples is, the more importance is given to showing that you have a representative set of samples.
And just to clarify here, the questioner was referring to CT as a clinical trial. So samples collected in a clinical trial that will be used for validating for the bridging study. What percentage should be available if it's not 100%? So that's what CT stands for.
Yeah, before you go, before you make a decision not to take a bridging study, but an external concordance study. Yeah.
Well, thank you very much for clarifying that. So I would like to say thank you very much for all those questions. We have reached the end of the question and answer portion of the webinar. If we couldn't attend to your questions, the team at IDDI will follow up with you after this presentation. Now, if you have any further questions, direct them to the email address that's on your screen, and you'll send that to dominique.grisard@iddi.com. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated, as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link, and also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, Els, Principal Statistician with the Consulting Services, and Everardo, Medical Director, both with the International Drug Development Institute, IDDI, for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. So thank you very much, Everardo and Else.
Thank you, Sonia. Thanks, everyone.
Bye.
Okay, bye everyone.
Bye now.